Our next presenter is Andrew Sedgewick.
Hi, thank you for having me. So the work I'm presenting here was part of a summer internship I did with 53 Genomics, which is a cancer genomics startup based in Santa Cruz. And for the rest of the year, I'm in Pittsburgh as part of the joint Carnegie Mellon PIP program in computational biology. So what I did was to work with the gene regulation part of the paradigm model. And paradigm is a pathway algorithm that was developed by some of the founders of 53 Genomics. So I'll give some hopefully brief background on paradigm so you can better understand what I did with it. So paradigm was actually presented at ISMB 2010, and it is a pathway model that combines pathways curated from a number of databases and uses those pathways to construct a graphical model which combines multiple omics data types into an integrated prediction of protein activity. So this is an overview of what we call the super pathway. So it's curated from these three databases and includes information like complex membership, gene family membership. The majority of the links are protein activation and inhibition, but there are also a lot of transcriptional activators and inhibitors. So this is a very simplified look at paradigm and factor graphs. So the subset of graphical models that paradigm is a factor graph. And in factor graphs, these ovals correspond to variables. And we use 3-state discrete variables that correspond to whether something is up, down, or the same compared to some control. And then the squares here are called factors, and they represent some function that connects the ovals that are connected to the square. So we use a conditional probability here. So each square corresponds to a conditional probability table. So for example, here we have, for every setting of the copy number variable, a probability of this, every possible setting for the expression state variable. And then in the end, our goal is an activity prediction, which we calculate based on this protein activity variable, and we call those integrated pathway levels, or IPLs. So here's what the paradigm model actually looks like. So for every gene, every complex, and every family, we have a graph that looks like this. And these shaded variables are where our data is linked to our graph. And these factors are just some small, just allow for like a really small amount of experimental noise. And so the way all of these are connected is through these regulation nodes. So the active nodes here are connected to regulation nodes based on which genes are annotated to be connected in the pathway. And the way we model complexes is just a noisy OR function of all the genes involved in that, noisy AND function of all the genes involved in that complex. And then gene families are a noisy OR function of all of the genes involved in that family. Okay, so what I worked a lot on was how does this connection through the regulation node actually work? So in original paradigm, or paradigm 1.0, this was simply a vote of the activation and inhibition signals that were coming into this variable. And in this new work, we're trying to model this regulation more rigorously. And so what we're modeling is the regulation variable which in these graphs is y, given the states of all the regulators that are coming into it. And we looked at two models, which we call a codependent model, where all of the regulators are connected to the regulation node through a single factor, and an independent model, where each pair of regulator and regulation node are connected through their own factor. And just terminology, I usually end up calling these parent nodes. So if I say parent node, that's what that means. And I usually call y here the child node. So the codependent method is really kind of a brute force approach. And it's kind of a problem you see a lot in machine learning where so you want to predict you have a lot of data, the parents, and you want to predict or classify some label, y. And so your first idea is, okay, maybe I'll just make a huge probability table that has every possible setting of the data, and I will figure out the probabilities and then use that to make my prediction. But you very, very quickly run into the problem that your table is exponential in the size, in the number of variables that you're looking at. So it gets so large that you won't ever observe the states, a lot of the states in your table. So you don't have nearly enough data to learn the parameters. And so for us to even implement this, we had to actually use an approximation that, so we used an approximation that limited the table to having 5 parents. And here's an example of what it would look like for 3 parents. We just split the factors. And that was necessary just to be able to run it. Otherwise, the table was like so large that it would eat all our memory. And right, another problem is with these huge tables, it's not even clear how to interpret something that has such high dimensionality in an intuitive way. So then, because of all these problems, we also looked at an independent model. And here we're essentially making a naive Bayes assumption of, which is that all of the parents are conditionally independent of each other given the child. And this model is nice because it drastically reduces the parameter space. Now it's linear in the number of variables we have. And so on the almost 2,000 TCGA tumors that we ran this on, that ended up giving us, on average, 200 data points per parameter. And because we have parameters for every link between parent and child, it's really easy to interpret this. And we can calculate our probability of the child given the parents by simply multiplying together all of our separate parent-child links. So here are a couple details about the implementation. So we had to do initializations of all our probability tables. So in the codependent model, we use the same voting procedure as was used in the original paradigm. And in the independent model, we initialize the tables based on the annotation of that link in the pathway. And so here would be an example of an activator. So when the regulator is down, the regulated variable is down, and so on. And I'll show a lot of these 3x3 tables. So you just remember that this positive slope means it's an activator, and this negative slope means it's an inhibitor. So the learning stayed the same. We're just using expectation maximization as implemented in libdi. And the outputs, so as before, we have, sorry, that should be IPL. We have an IPL for every entity. But now we also have these link parameters, which we can also do analyses on. And we have link parameters for every patient, but we can easily combine these into group level link parameters. So to kind of figure out what our link parameters meant, we had to do some statistics. So we determined link strength by looking at, by doing an independence test. So just testing if the joint probability is equal to the product of the marginal probabilities. And we use a G test, which follows the chi-square distribution. And so this is a sum over all possible settings of one variable x and one variable y here. And the elements of the sum are actually really informative because they tell us which settings are contributing to the dependence of the child on the parent. And so these are called weighted pointwise mutual information, or WPMI. And so we use these a lot. We'll either do something, a heat map like this to represent a single values of a single link. And also for a given setting, we can do box plots if we want to look across many link values. And then, so that gives us the strength of the link. And then to get the direction, we just do a simple Pearson correlation. As I mentioned before, we're using data from TCGA, and it's across 11 cancer types. And so we do some analysis on all of the data, and then we also pull out the ovarian cancer data to do a survival analysis. And in our analyses, these are the questions we were asking. So did we improve how useful the IPLs are? What, if anything, are we learning about links? And is what we're learning biologically relevant? So did we improve our activity predictions? Here, as I mentioned, we're going to use the survival data from ovarian cancer. And we have the IPLs produced by both old paradigm and our two new models. And what we do is just an unsupervised clustering of the patients based on IPLs. And then we look at the survival of those patients through Kaplan-Meier plots and statistics to see how well we're able to stratify the patients. So here are those results, and we find that our independent regulation model is able to significantly separate the groups of patients based on survival, while our original paradigm and our other model aren't able to achieve significant separation. So that gives us a good idea that we're kind of improving how useful our IPL predictions are. Now, we're really interested in these links because it's kind of it's an interesting new data to add to paradigm. So our inputs are our learned link parameters. And what we do is we calculate the WPMI for every link. Calculate the WPMI for every link, and then we clustered those WPMI vectors. We used a principal component analysis just for visualization, and we compared the directions of the links that we learned to their annotations in the pathway to see if they matched up. In addition, we used the results from the codependent model to test the conditional independence assumption we made with the independent model. So here is what the clustering looks like in principal component space. So we have 5 clusters, and they form a nice range along this parabola. So this is 1, 2, 3, 4, 5. And here is what the populations of those clusters look like relative to their annotation in the pathway. So clusters 4 and 5 are almost exclusively activators, and clusters 1 and 2 are almost exclusively inhibitors, and then cluster 3 is just in the middle. And we looked at the centroids of these clusters, and we actually found that they correspond really nicely to what our idea of a strong inhibitor all the way up to a strong activator is. And if you look at what the 2 principal components we're plotting here are, principal component 1 is modeling how the values in the corners, and then principal component 2, the up and down, ends up being the value in the center. So these two have a higher value in the center, and then up here there's a lower value in the center. So this is really cool because it's like a really nice gradient across inhibition to activation. So it seems like we're actually learning something. And we wanted to see how much our initial settings were affecting this. So we compared the initialization I described earlier to just using a uniform distribution of the parents and the children. And we found that we have a lot fewer significant links, but we don't have a lot of links, too many links that are switching directions. So we're still relatively confident in the sign of the links that we're learning. So here we're testing the conditional independence assumption made by the independent model. So it's a pretty similar g-test to what I described earlier, only now we're looking at the independence of every pair of parents given their child. And so at our starting point, we used that voting initialization, which is highly, highly conditionally dependent. So this plot shows the number of links that are affected by this conditional dependence. So this is how many links are conditionally, where the parents are conditionally dependent. So it starts out really high, but as we learn, our independence assumption becomes more and more valid. And then we also looked at, in these triplets, whether the directions of the signs were coherent. So here everything's an activator, so that makes sense, like all those directions make sense. But here, x1 is positively correlated with x2 and y, but then x2 is negatively correlated with y. So that's really ambiguous. That doesn't make sense, and it seems like one of those directions might be wrong. So we filtered by this coherence test and found that less than 5% of our links are affected by this incoherent codependence. So that gives us further confidence in our link directions. Okay, so we think we showed that we're actually learning something about the links, and now the question is what we're learning biologically relevant? So what we did here was we made a heat map and assigned a score to each link where the intensity of the color is based on the g-test strength, and the positive or negative is just based on the correlation. And so these are the most variable links across cancer types in TCGA. And here we're labeling by groups by the parents because they ended up clustering together. And so from this plot, it's easy to identify links that are differential across cancer types. And so we took these two groups of links because they had very little activity in most of the cancers and activity in only one or two cancers. So that was interesting to us. So we took these and discussed them with clinicians. So here's a more detailed view of the TAP73 tetramer complex. So this is the parent of all of these targets, and we're looking at plots of all of those links across the different cancer types. And we see that in ovarian cancer, there's a much stronger activation signal than in any of the other cancers. So P73, it turns out, is a P53 homolog, and the doctors we talked to mentioned that in high-grade ovarian cancer, there is an alternative form of P73 that's very active, so that could possibly be what we're seeing here. Yeah, similarly, we found this P-para-RXRA coactivator complex, which had a really strong activation signal in both glioblastoma and kidney cancer. And it turns out that both of these cancers are susceptible to the same chemotherapeutic fenofibrate, which actually targets P-para. So did we improve our predictions? It seems like we did based on our stratification test with ovarian cancer. And are we learning anything about the links? The gradient I showed, I think, is a really nice example that we think we are learning something about the links. And our independence test kind of support that we can be pretty confident in our link directions. And based on our discussions with clinicians and a number of enrichment tests that we did, I have time, so I'll go through this. We essentially looked at the, I didn't think I'd have time, but I'll go through it. We looked at the total top links across all of TCGA and did enrichment on the top 50. And we found that those were highly enriched for relevant Go and KEGG terms. And we actually compared that to just doing, just simply looking at the most highly correlated, just using a Pearson correlation of the mRNA expression. And we found that our links had much more relevant and much more significant enrichment than that simple correlation test. So yes, we think it's relevant, but as always, we could use more validation. So we have a site set up where you can see some of the supplementary data and register to run Paradigm through the web service at 5.3 Genomics. And I'd like to give a big thanks to Charlie Vasky, who originally developed Paradigm and helped me a lot with this project. And Steve Benz also helped a lot at 5.3. And thanks to my advisor, Takis Benos, and my lab mates. And I'm supported by a D32 grant. So any questions?
So it's a great talk.
Thank you.
So the Paradigm, the algorithm is mainly based on this gene expression analysis, correct?
Sorry, what did you say?
It's mainly based on the gene expression data.
Gene expression and copy number.
Copy numbers. So is there any plan to incorporate other data types like transcript binding and so on? I mean,
yeah, we're definitely actively working on incorporating microRNA and methylation. Because we would be really happy to get that working well, but we haven't gotten that working well yet.
So Paradigm trusts the super pathways quite a bit. Do you have any analysis showing how sensitive you would be to false positives or false negatives that the pathway curators made?
That's a good question. So the test with the flat prior gives us some sense that even without the annotations, we can still learn directions. But I think we should do a more directed analysis, maybe randomly flipping links and see if they switch back. But we haven't had a chance to do that yet.
Okay, thanks.
So I have a question about also the sensitivity or the robust of the algorithm. Such as if I just use gene expression or I use both gene expression and copy number, are the results similar?
Sorry, what was the question?
So I adjust the robust of the algorithm, such as if I just use gene expression and input, and use both gene expression and copy number and input, are the results similar?
They should be similar. It works slightly better with copy number. I think it makes it less noisy, but you can also just run it with gene expression, and we do that from time to time.
Okay, thanks.
Thank you.